Well, thank you. And I would like to start by thanking the organizers for inviting me to participate in this event and for giving me the opportunity to come back to this beautiful city. So I'm going to present you a brief summary, and I will try to make it brief, about the work our group has been doing during the last 15 years on cannabinoids and cancer. So as you all know, cancer is a disease that is produced by the uncontrolled proliferation of cells that have mutations. And this process is accompanied by the generation of new blood vessels in a process that is called angiogenesis. This new irrigation of the tumoral masses is done to feed the tumors and to allow them to grow. And at the latest stages of the disease, we observed what we call metastasis, which is the generation of new tumoral masses at distant sites. So if this scenario is not bad enough for a patient, for a cancer patient, some of them have to deal with pain and with the side effects of all the therapies they take to treat their tumors. They include things that can be considered minor, like hair loss, but other that are serious, like anemia, constipation, pain, more pain due to the medication, toxicity to major organs, and so on. So we know from many years now that oncologic patients can benefit from cannabis medicines, and we know that some of them do very well using cannabis, not sativas precisely, but other cannabinoid combinations to treat their pain, their oncologic pain. And we also know that these compounds work very well in treating the lack of appetite, nausea, and vomiting that is associated to chemotherapy. But what our group has been working on has nothing to do with palliative actions of cannabinoids, but with anti-tumor effects. We think that cannabinoids, and we know that cannabinoids can kill cancer cells. So when one is dealing with compounds that one thinks that have anti-tumor potential, one have to demonstrate two things that are very basic, very simple. Here, okay. The first one is that the compounds work as anti-tumoral agents, that they kill cancer cells. And the second one that one has to prove is that the compound is safe. We have a saying in Spanish, you don't want to kill mosquitoes with a cannonball. So we need therapies that are safe. And safe means that they produce no toxic effects on healthy cells, on non-cancer cells, but also when we are working with cannabinoids, that they produce no psychoactivity. And I want to focus your attention on this question mark. I will explain you later why. Well, we started to work around this first question at the late 1990s, and we focused our attention on glioblastoma multiforme, which is a type of brain tumor, extremely aggressive brain tumor. And these patients are currently treated with surgical removal of the tumor, plus radiotherapy, plus chemotherapy. And besides this very, very aggressive approach, most of these patients die within 14 months after diagnosis. So when we started doing experiments with these glioblastoma cells in culture, which means cultivated in plastic plates, we observed what you see in these two pictures. On the left-hand side, you can see healthy glioblastoma cells, healthy tumor cells. And what you see on the right-hand side is the same cells after treatment with THC. So we see that they become detached from the plastic plates, round, and after a couple of days, they disappear. So this is the first clue we had showing that cannabinoids were producing cell death. We then moved to more physiological models of cancer, and we have used many, many different models, animal models of cancer during these years. We have always observed the same responses. I'm going to show you just a couple of examples. Here on the left, we used what we call xenografts. We generate tumors by injecting, in this case, human glioblastoma cells underneath the skin of immune-deficient mice. So as you can see here, the animals develop big tumoral masses. And here you have an example of the size of the tumors in the population of animals that we do not treat with cannabinoids. So as you can see, they create huge tumoral masses that are red. And this is an example of an animal that has been treated with THC. And as you can see, the size of the tumors is smaller, and also the tumors have a different color. They are not red, they are white. And this means that these tumors are not irrigated, that not only tumor growth, but also the angiogenesis process has been blocked by cannabinoids. And this animal model here in the right is a little bit different, a little bit a step forward. In this case, we generated the tumors, the brain tumors, not in the back of the animals, but in the brain of the animals, in the original location. And this white mass here, can you see the pointer? Yes? Okay. So this white mass here is the tumor, this section and in this section. And this animal on the right is the very same animal after cannabinoid treatment. As you can see, there is no sign of the tumor at all. Well, we are also interested in understanding in molecular terms what is going on inside cancer cells when they are activated by cannabinoids. And this slide summarizes more than 10 years of work. And what we know today is that what cannabinoids do on cancer cells is activating a cellular program that is called apoptosis, which in very easy words means suicide. So what we have seen is that cannabinoids, by activating CB1 and CB2 receptors in most cases, change the amount of certain proteins inside the cells with these funny names. Don't waste your time trying to read any of these names. It's not important. The take-home message is that all these changes converge in the inactivation of this protein here, AKT, which is the master regulator of cell survival and cell proliferation. So whenever our cells have active AKT, they survive, they proliferate. And what cannabinoids do is inactivate this protein and then activate these cellular processes that lead to cancer cell death. As you can see in this table, we have seen these events, activation of cannabinoid receptors, inhibition of AKT, and induction of apoptosis, not only in gliomas, not only in brain tumors, but in a wide series of other tumor types like pancreatic cancer, hepatocellular carcinoma, breast cancer, and many more. So some years later, we started a new line of research in the lab, and this time focused on another type of tumors, breast cancer in particular, and among breast cancer, because we use the same word to define many different pathologies that affect the breast, but among them we decided to focus on those tumors that are characterized by an overexpression of this protein here. An overexpression means very high levels of this protein that is called HER2. So these patients are currently treated with a drug that is called trastuzumab, which has clearly improved the prognosis of this population. But besides this, not all HER2-positive patients respond to this treatment, and most of the responders end up developing resistance to the treatments. So we thought that this niche still needed alternative treatments. So to study the effect of cannabinoids on HER2-positive breast cancer, we chose a different animal model, which is called the MMTV neu mouse. And these animals have been genetically manipulated to overexpress this protein, neu, which is the equivalent in the rat to HER2. So human HER2 is called neu in the rat. So we manipulated the animals to make them overexpress neu, and as a consequence of this manipulation, these animals spontaneously develop breast tumors. So we don't have to inject cells anywhere because the tumors spontaneously arise. And they do it in a very similar way comparing to the human pathology. They do it after very long periods of time, very long. And in around 70% of the cases, these tumors generate metastases in the lungs, these white masses you can see here. So for all these features, these mice are considered a clinically relevant model of HER2-positive metastatic breast cancer. So what we did with these animals was to treat them during 3 months with THC around the tumor twice a week. And what we observed was first that THC reduced tumor growth. In this graph, we represent how the tumors grow with time. And as you can see here, if the animals do not receive cannabinoid treatments, the tumors grow exponentially. But if the animals receive THC, the growth is dramatically reduced. But this was not the only thing we observed. If you go to this table with me, you will see that THC also reduced tumor generation. Mice have 10 breasts, and each one of them is susceptible to generate a tumor. So we can have animals with 6 or more tumors. And what we observed was that in the control population means the animals that have not received cannabinoids, we found 40% of the animals with 4 tumors or more. And when we studied the THC-treated population, 50% of the animals have just 1 tumor, and we didn't see any single animal with 4, 5, or 6 tumors. So THC was reducing not only tumor growth, but also tumor generation. And THC also reduced the percentage of animals that develop lung metastases, which is very important. This is the the percentage of animals with metastases in the vehicle population. And as you can see here, this number is reduced, statistically reduced, when animals were treated with THC. But all the results I have shown you so far were obtained from animals. So we still didn't know, or at that time we didn't know if human cells could also, breast cancer cells could also respond to cannabinoids. So we started to use human adenocarcinoma breast cells, and we challenged them with THC, and we observed that cell proliferation, cancer cell proliferation, was also reduced. Not only when we grew the cancer cells in plastic plates, but also in animals. In this case, we used the xenograft model I mentioned before, injecting cancer cells underneath the skin in immune-deficient mice. And we treated the animals, in this case, orally, trying to mimic the route of administration that we think is more similar to what patients should do. And what we observed was that this white line represents the tumors that have not received THC, and we see a huge growth again. And when we treat the animals with THC, the growth was again reduced. And we could observe that the mechanisms that we had described for gliomas were also produced here. We observed that THC was was blocking cell proliferation. And in this slide, we were staining cells undergoing proliferation with pink. We were staining this protein, Ki67, which is only expressed in cells that are undergoing proliferation. So as you can see, I hope you can see pink dots in the picture. So if you compare the number of pink dots in this tumor section, you will see that it's higher than here. So it means that in non-THC treated animals, we have more cells undergoing proliferation than in animals that have received THC. THC was also inducing cell apoptosis, and in this case, what we are staining in pink is cells that are undergoing this cell suicide, and we observe an increased amount of these cells. And THC, and this is very interesting, was also inhibiting tumor angiogenesis again. What we are staining in this case is blood vessels within the tumors in green, and the picture is not very good, but we measured the amount of blood vessels in the THC-treated animals, and we observed that this number was decreased again. So this is just a brief summary of results suggesting or indicating that cannabinoids do work as antitumoral agents, at least in our preclinical models of cancer. The second question we have to ask is if cannabinoids are safe. And we think they are safe from the preclinical perspective and also from the clinical perspective. I'm going to show you a couple of examples of experiments we did to try to understand if the effect was produced not only on cancer cells but also on non-tumoral cells. And the answer is no. Here in the first panel, what we did was to challenge different cells with cannabinoids. The blue and green bars correspond to cancer cells, and the yellow bar correspond to, in this case, human mammary epithelial cells, breast cells, healthy breast cells. And as you can see, all cancer cells, when treated with cannabinoids, decreased their proliferation, they died. But this was not observed in healthy cells. Whenever we use a cell line, a cancer cell line, we always check that the non-tumoral equivalent is healthy. And what we have observed so far is that the healthy cells do not respond the same way they do, the same way as cancer cells do. So this is just an example. This is another example. In these experiments, what we do is taking a picture of the amount of DNA the cell cultures have. It's what we call the cell cycle. So when we observe what was the effect on the cell cycle in cancer cells, these two panels, we observe that this is the profile, this is the fingerprint of a healthy cell. And when we treat these cells with THC, we observe a change. If you compare these two graphs, we observe the appearance of a peak here. This one corresponds to cells that are dying. And we observe also a decrease in this part here, as you can see comparing these two parts of the graph. And this part corresponds to cells that are undergoing DNA synthesis, cells that are ready to proliferate. So THC in cancer cells is decreasing the number of cells that are undergoing proliferation and inducing cancer cell death. And if you compare these two panels, and here we are using the same lines, the same cells, healthy cells, than in these experiments, you can see that THC produced no changes in this cell cycle fingerprint. So again, these data were telling us that the effect of cannabinoids was selectively produced on cancer cells. And one more example, we have demonstrated that in order for THC to produce cancer cell death, the amount of this protein, JUND, has to be increased. And this is what you can see here. This is the amount we find of this protein in control cells. And when we treat them with THC, we increase the amount of this protein. And we don't see this effect when we treat non-cancer cells with cannabinoids. We, of course, whenever we do an in vivo experiment, we always check for signs of toxicity in major organs, lungs, liver, spleen, everywhere. We have never found any sign of toxicity. So all these results, again, suggest that the effect of cannabinoids is produced on non-cancer cells, is produced only on cancer cells, and that they do not produce any other toxic effects. But we're working with cannabinoids, and whenever we work to the medical community, they are very concerned about psychoactivity. So what we tell them is that we can avoid psychoactive effect by two different strategies. One of them would be targeting CB2 receptors specifically. The psychoactive effects of cannabis are produced by activation of CB1 receptors, of the CB1 receptors that are expressed in the brain. So one strategy to avoid this psychoactivity would be targeting CB2 receptors that are not expressed in the brain. So we have observed that at least in breast cancer, the anti-tumor effect of cannabinoids is produced also by activation of the CB2 receptor, the non-psychotropic CB2 receptor. And for example, we have observed that this compound, GWH133, produces exactly the same anti-tumor response than THC on our MTV-new animals. And we got the same conclusion from this other experiment here. In this case, what we did was to block CB2 receptors. So what we think is that the action of THC is produced by CB2. So if we block CB2 receptors, we should be able to not see the effect anymore, and that's what we observe. So in one of these lines here, you will see the behavior of vehicle-treated animals that did not receive THC treatment. this line corresponds to the animals that received THC treatment, and when we block CB2 receptors, when we don't let CB2 to be activated by THC, we lose the anti-tumor response. Okay, another strategy to avoid psychotropic effects would be to use another cannabinoid devoid of psychoactive actions. And a very good example is cannabidiol. So we have tested cannabidiol in our models, and we have observed that it works as efficiently as THC in killing cancer cells. CBD is represented here in the yellow bars and THC in the green bars. And as you can see, both compounds decrease the viability more or less in the same manner. This is in vitro experiments. We have also tested the compound in vivo. And again, we observed a very similar response. And I want to go back now to that question mark I put when I was mentioning psychoactivity, because we have to keep in mind that we are working with cancer patients. I mean, not we, but we are thinking about cancer patients. And we should reconsider maybe, or at least keep in mind, that the side effects of cannabis may be considered side benefits, as one of my best friends says. So the side effects of the psychoactive effects of cannabinoids include things like euphoria, feelings of well-being, relaxation, stress reduction. So we should keep this in mind and talk to the doctors and say that, well, some psychoactivity may not be that bad. Okay, and returning to the idea that cannabinoids are safe, we have a lot of information coming from clinical trials. There have been thousands of clinical trials, hundreds probably of clinical trials already performed. Here you have the numbers of the ones I found already completed. This was done 3 months ago. And we have almost 318 trials completed with Sativex, 86 completed with Marinol, 39 completed with CBD. The numbers are probably bigger today. And none of them have shown any effect that suggests doctors that they should not consider these compounds are safe drugs, safe medicines. So we have compounds that work, at least in preclinical models, as antitumoral agents that are safe. So what do we do next? Because we want patients to use these medicines, or to be able to use these medicines. Well, if we want to know if cannabinoids work as antitumoral agents in human patients, we have to try cannabinoids in human patients. So we did this a few years ago, in 2006, in a very, very small pilot clinical study with just 9 patients, very small population. It's very difficult to extract solid conclusions from this size. But still, we obtained very interesting results. So the primary endpoint of the study was to study the safety and the life quality of patients. But besides that, we took a look at survival, tumor growth, and effects on tumor markers. And this study was conducted in, I didn't say that, glioblastoma multiforme patients. And they were treated with THC. So the patients that were enrolled in this study were GBM patients that have undergone first surgery, the standard treatment. They suffered a recurrency, and at the time of the second surgery, a catheter with a pump was inserted inside the brain. And the THC treatment was prolonged until disease. And what we observed was that THC produced no psychotropic effects, that THC reduced tumor growth in some patients, and increased also length of survival in some of the patients. And THC did this as effectively as temozolomide, which is the standard therapy for this kind of patients. And something that was also very important for us, we observed that THC decreased the proliferation of cancer cells in these patients, that also blocked angiogenesis, and that increased apoptosis I have been talking for half an hour already, and I have mentioned this a couple of times. And what these results were telling us is that our preclinical models are valid. What we see in our animals has to do with what is seen in patients. So we are convinced that more clinical trials have to be done, of course. And there are two, or at least these are the two I know that are currently ongoing. There is one clinical trial that is being performed in the United Kingdom. They are studying whether GBM patients can benefit from the combination of temozolomide and Sativex. Patient enrollment, as far as I know, has been completed, and they say they will release results by the end of 2016. The other clinical trial I'm aware of is this one that is being conducted in Israel, in this case with pure CBD and with patients with solid tumors in general. And according to this website, the study started in 2014, and we will be seeing results by mid-July, by mid-2015. But we think that there are other sources of clinically relevant information as well. So we all know that cancer patients are already taking cannabis for the management of their diseases. But unfortunately, we are wasting all that information because we don't know how much THC they are taking, how much CBD they are taking. We don't have that information. Nobody's doing a serious follow-up of all these patients. And I want to give you an example of how things should be done, or how I think things should be done. These people are on CELDAS, which is run by Mara Gordon and Stuart Smith. Mara will be talking tomorrow. I encourage you to come and see her presentation. You will learn a lot. Well, what these people are doing with patients is, first of all, working with high-quality products. They have organically grown products. They know the composition of every single batch of products they give to the patients. They have a extremely highly controlled patient intake. They do personalized treatments. They see what the pathology is. They take into account things like age and previous use of cannabis and a lot of things that I cannot even remember. And they do a patient follow-up. So with all this information, they are able to provide enormous amounts of clinically valuable data that they will be analyzing and sharing with the medical community. So just to summarize, I'm convinced we have very strong preclinical evidence showing that cannabinoids work as antitumoral agents, that they are safe, and don't forget that they have additional advantages for cancer patients. They have analgesic properties, they have orexigenic properties, they are antiemetic. So we will be doing our job. We will be doing more preclinical science to try to answer pending questions. Like, we want to know more about the mechanism of action. The more we know, the more powerful we are in fighting against cancer. We want to know if these compounds combine well with standard chemotherapy. And we will do our best to force the medical community to do more studies, clinical studies. Observational studies probably at the beginning, but standard studies as well. And just to finish, I want to thank all the people that have been involved in this work during all these years, all the people in the lab, and especially the people that work on the breast cancer team. Manuel Guzmán, some of you know him, Eduardo Pérez Gómez, Clara Andradas, and Sandra Blanco. And thank you all for your attention.
Máme prostor pro dotazy, jestli se chcete Kristýny Sanchezové něco zeptat.
Okay, here's the question. Which way of administration would be the most...
I can hear you.
Oh, sorry. And now? Better? Which way of administration would be the most suitable for the cannabinoids? Cannabinoids dissolve well in fats, not in water, so they may not reach the tumor through the bloodstream. As we know, it is not always possible to deliver them directly to the tumor, for example, through a catheter, as Dr. Guzman did in his clinical trial that you mentioned. Do you think that liposomes could be useful, for example?
No, I would say oral administration would be the right way to go. Sublingual administration probably, but the clinical trial we performed in 2006, I wouldn't do it this way today. Whenever we think about cancer, we have to think about a systemic disease. So when I think about treating cancer patients, I would think about giving them the medicine through the blood system. So that would be my suggestion, and I think that's the best way to reach the tumor wherever it is. You don't always have access to the tumors, and we are not thinking about implanting catheters inside the brain of people. That's not the way to do it, because you also have to consider that most of the tumors have the ability to metastasize, and you don't want to lose track of these cells that are traveling around and generating new metastases in other places. So I would go systemically, and oral administration would be probably the best. Yeah.
Hi, I'm wondering if you could clarify the difference between CBD that comes from industrial hemp and CBD that comes from a medicinal marijuana plant, and if there is a difference, which one is more effective for the tumor?
Okay, well, CBD is CBD, no matter the origin of the molecule. You have a number of carbons, a number of hydrogens, a number of double oxygens or whatever, and the molecule is the same. But if you're working with extracts, the CBD-rich extracts that come from hemp have nothing to do with the ones that come from cannabis. You have to consider the rest of the accompanying compounds. So the molecule is the same, but the molecules that go with hemp or with cannabis are different.
So in your tests, what CBD did you use?
We used isolates, so pure CBD.
From the hemp or from what?
Well, they were
synthetic then, not from the plant.
Synthetic.
Okay, not from the plant. But if you were using it from the plant, what would you use?
We will use it. I mean, we will do some tests in the short-term future. We will be analyzing the effect. We will be comparing the effect of pure CBD, synthetic CBD, with the effect of CBD-rich oils that come with terpenes and a lot of other compounds and other cannabinoids also. So that's something we want to know. We want to know if CBD alone or pure CBD is better than extracts. We don't think it is. We think that the more we have from the plant, the better will work. But we are scientists, and we don't work by intuition. I want to be able to show you results, and that's what I hope I will be doing to do next year. So,
thank you.
Další dotaz?
Thank you very much for your presentation. Considering the synergistic effects of the whole plant, do you consider in any next future to study anything on the whole plant extract rather than single compound?
Yeah, we are starting these experiments right now in collaboration with other people. So we will be studying extracts from plants. Some of them rich in THC, some of them rich in CBD, and making combinations and trying to find out which one is best for each type of tumor. Because what we have seen is that, for example, breast cancer tumors respond better if you have more CBD. which is not the case for glioblastoma tumors. So we will try to see which is the best combination mixing these rich, THC-rich and CBD-rich oils.
So, sorry, so you're always considering decarboxylated extract. I'm just wondering because I'm very interested on the non-decarboxylated.
We know that if you don't decarboxylate the oils, you have a lot of THC-A, CBD-A acid forms, and they have their therapeutic value. But if you want to fight against cancer, you have to have high amounts of CBD and THC, the decarboxylated forms. So we may study as well what THC-A, CBD-A do on cancer cells, but we know that THC has to be there and that CBD has to be there too. So, well.
Hello. I want to ask you, you mentioned the pancreatic cancer cells. You mentioned pancreatic cancer cells experiments.
Are you asking if we have tried in pancreatic cancer cells? Well, we have not directly, but other groups have. And the response seems to be the same. THC induces apoptosis, kills cancer cells by inducing apoptosis. Yeah. And what we know from patients is that they respond as well.
Hello. You mentioned breast cancer cells as well as brain cancer cells. Have you highlighted cancer cells that are more susceptible?
No, that's not the reason. The reason is that our group started to work with brain tumors, so we know a lot about them, and then we decided to work on breast cancer cells. But we are not the only group in the world working on this, of course. And we have also worked with skin cancers, with hepatocellular carcinoma, and they seem to respond as good as the other types. I mean, there is no one response. Some of them respond, as I was saying before, respond best to THC. Some others respond best to CBD. Some others to 1-1 combination. So we have to find out which is the best ratio for each type of tumor, and within types of tumors for each individual patient. Yeah.
Nějaký další dotaz?
Jo, tady jsme slyšeli o rakovině prsu a slinivky, ale jestli je to obdobné jako u rakoviny prostaty nebo konečníku. Ty výsledky jsou obdobné nebo jsou odlišné?
Well, what we and other groups have observed is that in most cases the response is similar, meaning that in most cases the effects are produced by activation of cannabinoid receptors, the inhibition of this protein I mentioned, and the induction of apoptosis. So the difference may be who is the trigger of these responses. If THC, in the case of glioblastomas, for example, we have seen that THC is the most effective one. Or CBD, in the case of HER2-positive cancer, we know that CBD works at least as good as THC, but the final cellular response seems to be the same. So it's cells committing suicide in response to cannabinoids.
There is any kind of tumor which don't respond?
we have never seen a tumor not responding or a cancer cell line not responding to cannabinoids, and as far as I know, no other groups have found that. No.
Thank you.
Nějaký další dotaz?
Hello, Dr. Sánchez. Can you tell us if there is an ongoing study with the Complutense University at the moment, or soon expected to happen? Please. Thank you.
So you want me to talk about the medical cannabis bike tour? Okay.
Yeah, if you can highlight that for a second, please do so.
Well, I'm the one that coordinates the breast cancer group in Dr. Guzman group. So my colleague Guillermo Velasco is the one in charge of the glioma studies. And as you very well know, they are trying to move to a clinical trial with glioblastoma patients. And the money will come from the medical cannabis bike tour, as it was presented this morning. So we hope that this clinical trial will start in the next few months, probably spring. Now, probably you have more information than me with this. So they will be starting a clinical trial in Spain, in Madrid, with, I don't remember the number of hospitals, but it's at least 3 of the main public hospitals in Madrid, and with patients with GBM, glioblastomas, and away from the big pharma companies.